following paralysis with use of brain-controlled stimulation of muscles. It's a 2012 article from Nature, and I found it on the Nature Neuroscience blog, cleverly named Action Potential. The article is pretty short and written by one of the editors of Nature. And it got me pretty interested because it's kind of a futuristic sounding thing. There's been a lot of other work done previously with brain-machine interfaces in Star Wars, as I discovered when I was doing this. I didn't know it before. Not a big Star Wars watcher, but Luke lost his arm and had a robotic arm, and he would think what he wanted the arm to do, and it would do that. And that's actually being done now. So the picture up here, this is actually from a study that came out after this one, just a couple days after, but a researcher at Dartmouth was able to hook up some electrodes to the brain of this woman, and she was able to think that she wanted to take a drink of water, and the robotic arm moved the water to her mouth. She took a drink of water. So that's pretty awesome. I guess I am on the timer. Moving on to this article, every year there are over 130,000 spinal cord injuries, and a lot of paralysis comes with that, and over half of those injuries are above the 6th cervical vertebrae, which means that there can be paralysis of all 4 limbs, which is pretty significant. A lot of these patients talk about that if they could have some ability back, it would be the ability to grasp items. It's pretty frequent motion in our daily lives. There has been some research done with functional electrical stimulation, which is a technique similar to the robot hand, but instead of using a prosthesis like that, it uses what they call neural prosthetics, which is using your own muscle and function and the limb that you still have to do the movement. But it's been pretty limited so far, with just a couple of motions and not much control of strength of the motions either. And it uses just residual motions that the patient still has, residual ability, in order to make those movements. But the field is moving really quickly with brain-machine interfacing, BMI, and there's a lot of possibility with it for giving back a lot of motion and function to paralysis patients. So the article focused on the primary motor cortex, which is right in here. And the motor cortex can be broken down into areas for different parts of your body moving. So they were focusing on certain muscles that they specify that I'm not going to talk about, but certain flexor muscles in your arm. So the primary motor cortex they abbreviate as M1. And they hypothesized that using the functional electrical stimulation would be much more effective for basically bypassing the spinal cord injury in paralyzed monkeys. It would be more effective than previous things that have been done, though they don't compare in their study. So they used 2 monkeys in their study, just 2 subjects, and the monkeys had been trained with this weighted ball activity where they were taught to lift a ball from the bottom of the chute and put it back down the chute. And then they were implanted with electrodes in 2 areas. First, they were implanted in the brain, in the primary motor cortex, in the area specific for those hand motions. And then they were also implanted with electrodes in their arm below the elbow. After that, they took recordings from the primary motor cortex using the first implanted electrodes, and they got some baseline data of what brain activity and neuronal firing activity happened when the monkey did the ball drop task. And then they use that as a predictor, which I'll talk about in a minute. So in between here, they would deliver a nerve block, which was a lidocaine injection, and it would block the radial and ulnar nerves at the elbow, which was basically stimulating the effects of a C5 or C6 paralysis. And they would wait for that to go into effect, and then they would stimulate the effect. They would use the functional electric stimulation on the muscles in the arm to stimulate the neuronal firing activity that was going on in the brain. I'll explain this a couple more times. But basically, they're trying to bypass this roadblock of paralysis. So there's firing going on in the brain that's saying, pick up the ball and put it in there, but that signal isn't reaching the nerves in the arm because there's a big roadblock here. And by using the electrical activity that they've seen in the brain and delivering it right here, they just jump over to here, and then the arm is able to move. So this is what I was talking about before of predicting what the signals look like. The blue is the actual signal, so that's what they're detecting in the brain from the primary motor cortex, and the red is what their functional electric signal is able to predict is happening, and this is all done with an electromyograph EMG. So this is from a 20-minute session. There's a lot, and these are the accuracy in the different areas. They had electrodes implanted. They had 5 electrodes in the arm, so that's the accuracy for those. So this is the really awesome part. I'm going to show you a video of them doing the study. And there are 3 parts of it that will make it very clear to you what's going on. The first part is before they've delivered the lidocaine nerve block. So this is just normal monkey doing the trained activity. Then it's going to say FES is on, and that's when they've started the stimulation. They've received the nerve block, some time has gone by, so they're effectively paralyzed. Parts of them are paralyzed. And they're receiving electrical stimulation based on the patterns that have already been predicted in real-time predictions. And then it'll say FES off, and that's when they've turned off the stimulation, and so they're paralyzed, but they're not getting the stimulation. So that would just be like your typical paralyzed monkey. With the stimulation on. And after the nerve block has been delivered.
That is blocked?
Yeah.
The nerve is blocked?
The nerve is blocked. And now the nerve is still blocked, but it's not getting the stimulation. There aren't any outlets.
We couldn't find anything closer. Crisis averted.
All right. Great. So, I think that's pretty awesome. So you can see from this, the main thing from it is that the grasp was remarkable with the stimulation on. It was effective. It was not very different from before the block, and it was reliable. They were able to do the task very well. During what they called the catch trials, when the block was still on but they turned off the stimulation, they couldn't do it. They were able to see that the firing activity, they were putting in a lot of effort. It doesn't show up quite as well on here, but there's more, the activity is darker here. So there's more firing going on there than there was when the electrical stimulation was occurring. So they're trying really hard, but it's not working. They can't do it. And you can see on the graph here, there's no black line. There's no stimulation occurring, but the activity is still going on as being read by the electrodes in the primary motor cortex. The success rates. This shows the success rates in just 2 of the trials. The green is with the stimulation on, and the blues are without the stimulation on, the catch trials, where the block is still there. And 5% and 0% success rates when the stimulation was not on, 80% and 90% success rate when stimulation was on. And D shows the overall success rates very significant. The video is pretty glaring in the results. In addition to having just a higher success rate and being more able to do the task, they also did it with greater speed with the stimulation on than they did without the stimulation. They got through more trials when they had the stimulation on than they did when they had no block, which is interesting. I'm not sure what to think of that. They didn't really discuss that very much. And then you can see it was not effective at all with the catch. So with FES turned on, it was really effective. They could do the task at a high rate. Another thing they talked about in the paper, which I'm not going to cover too much, they also looked at grasp force, and so how effectively they could squeeze the ball. They had a task where they had to squeeze the ball in order to keep a target on something. Only one of the monkeys did that, but he was more successful at squeezing the ball when the stimulation was turned on than he was when it was turned off. So that's a big leap forward from a lot of other studies which haven't looked at force. They've only looked at movement. Conclusions. The model of paralysis that was used in the study may not have been so realistic. They didn't. The monkeys were not actually paralyzed, and so didn't have any denervation, which is loss of nerve supply, or spasticity, which is stiff or rigid muscles. And so they had very healthy muscles that were being used. And also the extent of the paralysis. They mentioned this limitation in their paper, that they didn't paralyze all parts of the forearm. And they were saying that was a limitation, but it was also a reason why it could be very useful for the most severe paralysis patients, which I didn't quite understand how that worked. But they only used 2 subjects, 2 monkeys, so expanding that, and they didn't manipulate the ball at all. And they talked about how using the stimulation would be a great exercise for patients in terms of psychological health, of being able to achieve this. This is talking more about human patients. And also more study with more electrodes and what the possibilities would be if you just expanded it. So finally, this is really cool technology. There are a lot of possibilities, and that would apply to a lot of people. More study with more electrodes and with humans eventually would be great. Thank you very much.
So the timing was perfect. Questions? We have 5 minutes for conversation.
Yeah. They talked at all about like a comparison of the number of electrodes that it would take in a human or like a fully paralyzed limb. They used 5, but did they mention how many
They didn't, and I think that's one of the things is that the reason they're talking about doing this with spinal cord injury patients who it's not like 100% across the board paralysis. It's not all your nerves are completely destroyed. And so there are more possibilities with I think the possibilities are different with this than with someone who has lost a limb and has a prosthesis, because you don't necessarily need as many electrodes. But they didn't really talk too much.
So in the experiments, was it just the monkey's hand that was paralyzed then?
Yeah, the injection was delivered in here to the radial and ulnar nerves above the elbow.
Okay.
So it isn't paralyzing the entire hand.
Median nerve.
Median nerve.
If they didn't block the median nerve, there's another nerve that brings major impulses to the hand, and that's the median nerve.
So they just basically, they basically just blocked their ability to grasp, and that was the only thing they were testing for.
I think so. Yeah, that's my understanding.
Awesome. Got it.
So I'm not totally sure I understand the electrode and how it predicted the yes. I'm not sure about the either.
Okay, I think this is the most helpful one. So there are electrodes in the primary motor cortex, and so that's getting a signal of as the monkey is doing the task. So reaching for the ball and putting it in, the electrodes are reading what's going on in the brain, and that's what I showed you of the neuronal firing activity. And so showing that here, that's the data from there. And so that is predicting what the movement will be. You can read from that. Some people can read from that, that the hand will pick up the ball and put it in the tube. And so that's what the prediction is. And based on that, their decoder machinery puts in an electrical prediction stimulator. It puts a command into their stimulator, the functional electric stimulator. And that electronic code basically says how strong and for how long the stimulation needs to be to match what's going on in the primary motor cortex, and it sends that signal into the electrodes in the arm, into the muscle, and makes it happen. Does that make sense?
So basically, they're reading the brain, reading the electrical signals in the brain, decoding it, and turning it into the language that the muscles can understand, and then feeding that in themselves, not from the brain directly to the muscle, but from the machine to the muscle, to take the commands from the brain to the machine, and then the machine to the muscle. I mean, in this
bypassing
the nerve.
And the nerve, yeah.
And so this is the way a paralyzed person could end up moving muscles, because they can imagine moving their arm, if they can think about moving their arm, and the machine can decode that thought of moving their arm, then the machine can move their arm for them.
So does the electrode deliver an electrical impulse or like a neurotransmitter?
Electrical.
Electrical.
It's stimulating the nerves that are still exist. So that is one of
Is there?
the nerve is still there. Yeah. So this is one of the limitations of this approach, right? Because if someone becomes really paralyzed due to a spinal cord injury, their muscles start to atrophy. But then the idea is, well, you could just have a robotic arm. They could start to move a robotic arm.
Yeah, I was going to ask about that because it would actually be most likely more easier to move a robotic arm because we understand the language of a robotic arm better than the language of the biology of our own arms.
That's right.
So
Yeah.
Anakin's arm might
Yeah.
work a little better.
Yeah.
Actually, similar to that, I was wondering, do they mention at all, like, if they compared the data from the two different subjects, like if they were similar at all, or if they were
From the
basically.
two monkeys?
Yeah, basically what I'm after is this the kind of thing you'd be able to generalize to multiple people from one subject, or would each individual have to get their own readings?
Well, so two answers. The two monkeys performed very similarly, but their patterns aren't identical. So it has to be a custom program.
Plus,
for an
the
individual.
positioning of the electrodes
Right.
in the brain is so individual, so that the code that they're reading from the brain is going to be fairly individual. So fine-tuning that for an individual person is going to be tricky. Because you can't, if you've got a paralyzed patient, you can't train the machine, right? It's already paralyzed. Okay, one more question.
Okay, so if it's like essentially like a prosthetic sensory input, do you think like over time, if someone had been paralyzed for a while, not only because of the muscles atrophying, but because the neural pathway had been like physically extinguished, it wouldn't work, or it wouldn't work as well?
Well, I think that the atrophy of the muscles is a much bigger issue than the neural pathway, because one of the things I've been very excited about in reading a lot about is the plasticity of the brain, and there are incredible studies that are pretty sensational coming out about how much ability we have to recover there are studies with stroke patients 10 years afterwards regaining certain abilities. So it's like they're pretty sensational articles, but I don't think that's as big of an issue. They did mention briefly at the end that it's important to note that doing this wouldn't limit the ability of the brain to adapt farther. But that even with adaptation, it's like that's a lot of work for the brain to do with all that damage, with the amount of blockage, and that having the electrodes is helpful to take the burden off of how much work would have to go on to overcome those.
Okay, let's thank Elsa again. So that was a little bit more than 5 minutes of conversation, just to give you a sense of how long a 5-minute conversation, what kind of content can be covered in a 5-minute conversation. I'll need to keep the time fairly focused on 5 minutes in order to get through the talks in the time allotted for the course, or for each class. I'll take the professor's final prerogative. When I first heard about this technique, I found out that the Department of Defense was funding a lot of the research on this, and I thought I was concerned that might be insidious. That the Department of Defense is trying to work out brain-machine. Well, you can imagine what kind of machines the Department of Defense is interested in, brain-machine interfaces. But the Department of, in their defense, no pun intended. The Department of Defense is very interested in spinal cord injury. A lot of soldiers get spinal cord and brain injury, so their interest in this kind of technology is not particularly evil. It could be quite appropriate.
I actually looked stuff up like 3 years ago, and they actually have, which is really kind of nerdy of them, but they actually have a similar to, which is ridiculous sounding, similar to an Iron Man suit kind of thing.
Oh, really?
They're trying to build, which I'm surprised they're even trying, because it seems just kind of
Great.
ridiculous.
Yeah.
But
so that's a brain
so maybe
machine.
yeah.
So that wasn't the kind of brain machine interface I was thinking of.
In Avatar.
Avatar, right?
Yeah.
Yeah.
Just get in and I had to think.
Right.
Yeah.
But you can imagine, like a brain-machine interface. One of the limitations of fighter pilots is the reaction time. You're flying so fast, the human reaction time becomes quite limiting. Well, you can imagine if you can remove the muscles and your brain is talking to the plane, that could give you a competitive advantage. I don't think that's where the Department of Defense is only interested, but I do think that is part of what their interest is in. Seth, last comment.
a more benign version of that, and heard that they're also looking at like brain-controlled computer interfaces.
Uh-huh.
It's like basically giving people who are really thoroughly paralyzed the ability to control, like, just basically a home computer or something like that.
Yeah, right. Okay, so what I would like to do is collect your comment sheets. This will be Elsa's feedback. She's learning, how to do this well too. I mean, that was an excellent presentation. You can probably see some of the things that we're looking for. There were some of the things that I felt Elsa could have done better. For example, walking us through the complicated data figures. What was plotted on the y-axis versus time, obviously on the x-axis. That there's room for improvement there, but that was an excellent talk. That is the kind of talk that you should strive for when you give your presentation. So I'll collect your comments. Elsa will receive your comments, and it will be part of her improvement. And when you're done, I'll see you next week.